All right, thanks, Jem. Thanks for the invitation and for everyone who is here. Um, oh, I thought I should have a discussion on arsenic and mercury in shallow submarine hydrothermal systems. And this is part of work that I'm doing linked to the dynamics of arsenic cycling through Earth history, going back to the Precambrian. And I've been very interested in using this sort of systems as some kind of analog to interpret what happened in deep time. Now, as you see here, this work is not, I mean, I can say this, I'm talking on behalf of Yu Cheng. She did a lot of the work that I'm going to present here, which is mostly metagenomic work. But there are others that are involved: Magnus, Noah, and Nicole, Stephanus, and Ariane, who are all in the field for the sampling of the sediments that I'm going to discuss in this work. So bulk of the work is based on these hydrothermal systems in Greece. Now, why arsenic and mercury? To start off with, I think we're very much aware of the fact that arsenic is a toxin to start with. There's been a lot of exciting movies, if we can put it in that way, where arsenic is used to as a toxin, and then it starts a whole mystery. We know that presently arsenic is causing a lot of problems to a lot of regions on Earth. For example, in Southeast Asia, where arsenic toxicity is quite rampant, and it's all linked to geological processes where this arsenic is coming from getting into groundwater. Now, mercury on its own as well, I think the first time I really got to know about mercury was its use in thermometers, and then later in medical applications, way going back deep in time to treat diseases, which is something that's being, I think we know more now not to use mercury in the forms that they used to use for medical purposes. But yet this mercury presents a lot of problems today. For example, if you think of Japan, there's the Minamata disease, where mercury was released into marine system, and then it got biomagnified, and the consequences, became obvious in the people who consumed the fish. So understanding the biogeochemical cycling of arsenic and mercury, it's very important. And also trying to understand how that is linked to other biogeochemical pathways, because obviously arsenic and mercury toxicity is not limited to animals. It would go down to the microbial level. If we look at the distribution of hydrothermal system on Earth today, they're quite vast. You can see the mid-ocean ridges, the Iceland Arc, and systems like what we have here in the Hellenic Volcanic Arc, which is where I'm going to focus this work, but also back arc environments, back arc basin environments where you have a lot of volcanisms and submarine hydrothermal activity that releases a lot of trace metals that could be beneficial to life and essential nutrients. But at the same time, we know that at quite elevated concentrations, these trace metals can become very toxic. So obviously hydrothermal systems, they contribute to the nutrient budgets of the oceans, and at the same time, they can also be quite, as these are extreme environments, of course, they can also have a negative impact on the life that exists in this environment. So now, if you look at, so I've borrowed this table from this publication down here. It's a review of arsenic in hydrothermal systems and fluids. If we look at the case of arsenic in Iceland island arcs, for example, here, we find that we can get as much as 5,850 micrograms of arsenic in hydrothermal fluids. And according to these authors here, they say that this is about 3,900 times the concentration that you get in open ocean seawater. Now, that's quite a lot of arsenic, considering that open ocean seawater has about 1.7 micrograms per liter of arsenic. And if you move further to the ocean, mid-ocean reaches, this is the mid-Atlantic Ocean reach. This one here in the East Pacific rise, we can get concentrations as high as 80.5 micrograms per liter, and then in back arc basins, that high as well. So it seems as if generally the shallow hydrothermal systems do contain a lot more arsenic than the deep ocean hydrothermal vent fields. As I was saying before, these are really extreme environments. You see pH usually acidic, temperatures are high and variable. The depth as well. We do have different sorts of lithologies that are linked with these different systems. But what is kind of interesting is that the amount of arsenic that we find in these different environments is not tightly linked to the rock types that form the basins. Now, I used some of the data from the previous table just to look to see, what was the impact of, say, pH, temperature, and depth on the distribution of arsenic in the systems. What we see then is that the pH doesn't seem to have a great impact on the concentration of arsenic. Temperature, there's some sort of an inverse correlation, but what we see is that it's just this relationship, just about 30% of this relationship can explain this distribution. But it's very clear that as the temperatures drop, more arsenic it's present in the hydrothermal fluids. Similarly, if you look at depth, there's also an inverse correlation as well, not strongly supported. But again, we see that as you get to shallower depths, you start to get more arsenic in the fluid. So, which brings this very close to us, because the shallow hydrothermal systems that have been, or that we're going to talk about here, you can actually swim over them and see this diffuse hydrothermal activity occurring on the seafloor. And that means, what that means is that there's a potential for a lot of arsenic to be released from the systems into seawater. And that can then have an impact on local biodiversity. But is this actually the case? Now, if you look at mercury distribution, what we see from the study here, I'm just going to focus on the case of millers. There's probably a lot of other studies that have looked at mercury in other hydrothermal systems. But in the case of millers, we see that most of the mercury is associated with the pore water fluids, and very little is found in seawater. So what is happening here? Why is all that mercury being concentrated in the pore water fluid? That is of interest. So these other authors, they then went forward with the same system, and looked at the mineralogy and tried to target the mercury. Where is it in the sediments? They found out that a lot of the mercury is actually associated with pyrite, sulfide minerals, and cinnabar, which is a mercury sulfide. and that this occurs in association with other minerals like calcite, apatite, and other sulfide and sulfur minerals, including iron oxides. So it seems as if there's a system in place that's actually sequestrating the mercury when it comes out of the hydrothermal systems, locking it in the sediments so much so that very little is escaping into the water column. So what are the questions that I'm asking here? First, what dominates biogeochemical arsenic cycling in the shallow submarine hydrothermal systems? Is it arsenic detoxification, or is it acquisition of energy, or both? What are the principal pathways to mercury biogeochemical cycling? Is it methylation or reductive cycling, which confers resistance, or both? How are these processes coupled to other biogeochemical cycles or pathways, for example, sulfur and phosphate? And this is very critical because phosphate, as we know, is a macronutrient, and it's required by all of life as we know it. Then what we do here then, or what we did then, was to use comparative metagenomics to investigate the differentiation of this seafloor habitats into different ecological zones influenced by seafloor hydrothermal activities. And the goal here was to look at the distributions of different genes involved in different biogeochemical pathways, for example, arsenic detoxification, arsenic oxidation and reduction, mercury methylation and reduction as well. Genes involved in sulfur cycling, is it linked to their prevalence, for example, sulfur oxidation and sulfate reduction. And our focus, as I was saying before, is on Miller's Island. So what is happening here is that this is the African continent, the African plate. This is the Mediterranean Sea, that's the Aegean Sea here. There's the Euro-Asian plate, which is light comparatively to the African plate. So what happens is that the African plate is being subducted underneath the Euro-Asian plate. And what then happens is that we have this Hellenic trench right here. And there's the Hellenic Volcanic Arc, which includes Milos Island here in red, and there's Santorini here. It's intriguing because Santorini, this very popular holiday resort is actually sitting on the mouth of a volcano. And this is Crete here, and the Hellenic Trench is somewhere out here. And 100 kilometers away from Crete, we have this Hellenic Volcanic Arc. And our study site is here at Milos. And you will have, where you see these beautiful landscapes, shallow submarine hydrothermal venting occurring here, close to this. Close to the coast, and down here some pictures of gas bubbles, diffuse hydrothermal activity. These gas bubbles include CO2 and hydrogen sulfide, and you tend to have seafloor mats, white seafloor mats that are rich in sulfur being precipitated in where you have these gases escaping. So it's same thing that I'm talking about here. So what we did was basically we sent scuba divers down, and they sampled the system using push calls, along a transect that cut across the white mats, and there's brown reddish mats that are located just on the periphery of the white mats, and then the areas that were not in influenced by hydrothermal activity or visible hydrothermal activity, mainly sand as reference. And then we applied metagenomics using these various tools here. So there's this steps from sampling and DNA acquisition and the tools that we use to actually compile the data. Now, again, we differentiate three distinct zones, which we call the white cap site, the brown cap, and the reference site. Most of the analysis that I'm going to show you comes from the sites, from these locations that are labeled here, VM, V1, VM, V1, and so on. The reason being that we did sample down to 20 centimeters or more, but we could only obtain DNA that we could do any meaningful analysis with from this location. So the data is mostly for those locations. Using the chemistry, mineralogy, for example, arsenic 3 concentration, arsenic 5, phosphate concentration, different minerals, iron oxide, sulfur, and sulfide minerals. We did multi-dimensional scaling, and we saw it grouped the zones into three major zones, which we call zone 1, made of composed of this and this mats here, and then zone 2, which was basically the sediments beneath the brown mat. Then zone 3, which was the reference material. Now, this is what we see when it comes to the distribution of microorganisms or bacteria in the samples. So we go from W way out to the reference area. Here, this is the center where the hydrothermal activity is actually occurring, where you find the gas bubbles, and then the brown mats, and then the reference. What we see is that similar to the geochemical zones, biogeochemical zones, or geochemical zones that we identified, we also see that the bacterial population changes along that gradient. For example, we do have Campylobacter bacteria, which basically are sulfur oxidizing bacteria, dominating in the hydrothermal centers. And also we do have this chlorobia. It's quite prevalent in this area as well. As we move outwards, we tend to see that the delta proteobacteria or the gamma proteobacteria become more prominent. And as well, we start to see oxyphotobacteria, for example, cyanobacteria becoming more prevalent, although small relative to the other classes, bacteria classes, we do see that they appear. They're not prevalent in the center of the hydrothermal system, probably due to the extreme environments, the high temperature, and metals that are quite toxic being released as well, metal logs. When we look at the archaea, we see a similar trend as well. There's a change in the composition as we go from the hydrothermal centers to the reference material. We see, for example, that demococci is quite prevalent in this hydrothermally active center. And as we move outwards, the demococci disappear, and nitrososphere becomes prominent, and as well as other classes like low-care archaea appear here, including ordinary archaea appear in the reference material as well. So there's some sort of clear connection between the geochemistry that we're seeing I'm talking about, so the distance is about 12 meters. You can see this differentiation occurring quite quickly along that gradient. It was quite interesting because we found out that it was interesting to see a lot of diatoms in the system, really. They're also photosynthetic, obviously. And their distribution was widespread. It didn't matter whether it was the reference high hydrothermal activity area or not. They're quite prevalent. Now, we attempted to look at the to assemble genomes, whole genomes from these various samples. And what we found out was that the epsilon or the Campylobacter, obviously, they dominated in, as I showed previously as well, they dominated, we could obtain genomes related to this class in the hydrothermally active center. And as we moved outwards, that became very rare as well. And again, we see the gamma proteobacteria appearing in genomes related to the gamma proteobacteria being more prominent in the reference. We looked at the distribution of arsenic and phosphorus-cycling genes. The reason for this is that phosphate transporters, because of the similarity between arsenate and phosphate, arsenate tends to get into cells using phosphate transporters. And so this is really a problem for a lot of microorganisms, especially those that are existing in this kind of environments where arsenic is very high and can be oxidized to arsenic 5. The acquisition of phosphate can become a big problem. So we looked at the distribution of these genes, and what we saw was that, here, this genes involved in pumping out arsenic from the cells, as B, as C, as A, and comparatively to genes involved in arsenic oxidation, we didn't find any genes involved in arsenic reduction. This detoxification genes are widespread across all areas from the hydrothermally active centers to the reference. Of particular interest is this RC FOX pump, which has been dated and shown to have evolved way back in time, deep during the Archean period when the oceans were quite hydrothermally active. This gene is quite widespread in this environment, and even with quantitative PCR, we could see that this is the most predominant gene involved in the extrusion of arsenic from cells. We find phosphate cycling genes, for example, the PSTA, PSP, PSTB genes that are involved in high affinity phosphate uptake, meaning that these are really, really efficient genes when it comes to the ability to take up very tiny amounts of phosphate in the environment. And they can discriminate against arsenate pretty well. These genes are very prominent across all the sites, implying that there's a mechanism in place to actually find low phosphate concentrations and at the same time prevent the cells from accumulating large amount of arsenic within them. Looking at mercury-related genes, most of the genes linked with mercury cycling were those involved in mercury reduction, and also for the transportation of mercury in and out of the cell. We didn't find any genes involved in methylation, which is kind of interesting because from one of the papers that I showed before, the authors, they could not find any genes, or they could not find any, when they quantified the different forms of arsenic in this hydrothermal, mercury in this hydrothermal system, they could not identify methylated mercury forms. But what is intriguing is that we find most of these genes located in the reference. And you will remember that in the hydrothermal systems, most of the mercury seems to be associated with pyrite. It's precipitated and removed out of the pore water. So probably that mercury is not available. Meanwhile, it appears as if as you move away, the mercury becomes more available in the reference system, implying that you might, or the organisms living here, that require a mechanism to detoxify mercury to stay safe. We also find genes involved in carbon monoxide oxidation. Methane oxidation is very rare. Genes involved in methane oxidation were not predominant in this environment at all. Looking at genes involved in arsenic cycling, sulfur cycling, sorry, I'm in love with arsenic. What we see here is that we find a full set of genes related to the oxidation of sulfur. And these genes are mainly present in the hydrothermal centers, not in the reference. So sulfur oxidation seems to be a really important process here. And you remember, the data showed us that we have this Campylobacter bacteria, or Epsilon proteobacteria, which they are involved in mainly in sulfur oxidation, and it's been shown in the system before that they're quite widespread in these hydrothermal centers. We also find DSR, A and B, which are genes involved in sulfur reduction and prevalent mostly in the sediments underneath the brown mats, and sometimes in the reference as well. There's also genes involved in the RTCA cycle and Rubisco genes. We find them distributed across the system as well. Not going to go too deep into that right now, but carbon fixation, it seems to be really prominent here, and Rubisco is involved, and the RTCA cycle as well. It's prominent. Now, putting all of that data together, we try to come up with some sort of a conceptual model for how, let me see, come up with conceptual models for, for how arsenic and mercury, it's like the cycles in this environment. We know that most of this work is based on DNA. Probably to actually get a good idea, you want to do metatranscriptomics to actually quantify the genes that are active. But based on the distribution of these genes in these different environments, based on the microbial assemblage as we know it, and also from other works that's been done here, we think that most of the arsenic in this environment gets into the cells through phosphate using the through phosphate transporters. Once in the cell, we do have a C, which is used to reduce this. This enzyme is used to reduce arsenic to arsenic 3, which is then pumped out of the cell using these different FOX pumps. It's kind of intriguing because the cell has to go through this tricky process to transform arsenic 5 to arsenic 3, which is more toxic, before it can get it out of the cell. The reason being that arsenic 5 is very similar to phosphate, and the cell is trying to prevent losing its phosphate by co-extrusion with arsenic 5. So it has to reduce that arsenic. to arsenic 3, and then it's thrown out. So we think this is what is happening there. There's a little bit of methylation going there on there, arsenic methylation. There's some arsenic oxidation occurring. We could not identify arsenic reduction as a prominent process in this environment. For mercury, we think that this mercuric ion transporters they transport mercury into the cell or cells in the system, and that mercury is reduced and then sent out of the cell as using various transporters as well. Now, so what does this mean in terms of the whole ecosystem? So what we see then is that you have hydrothermal activity occurring. The fluids come up with arsenic, mercury, iron 2 sulfide is precipitated in conjunction with with arsenic and mercury, form these various sulfide minerals. What we think then is that in zone 2, you do have some of that pyrite is being oxidized, and there's recycling of some of that sulfite, and pyrite is getting precipitated. But then again, you get into zone 3, where we have iron oxides being most of the minerals we found here are iron oxides. That's where we start zone 2 and 3, we start to see mercury detoxification occurring, while arsenic detoxification is occurring in all zones. So clearly there's a strong connection between the sulfur cycle and arsenic and mercury cycle in this system. And we don't see a lot of arsenic respiration occurring. There's some arsenic oxidation. The system is mainly the microbial processes here involved in arsenic cycling are focused on detoxification, which makes sense given the amount of arsenic load that is they're exposed to. We could not detect biological mercury mutilation occurring there. There's strong reduction of mercury as a way to gain resistance to arsenic, to mercury toxicity in this environment. And there's widespread high affinity phosphate uptake, which again is connected to the fact that you have iron oxides forming there that actually remove some of the phosphate from the system. And at the same time, there's a lot of arsenic and arsenate as well. It also confers some resistance to arsenic toxicity. All right, that's it from me. Thank you.
Thank you, Ernest. That's very, very good. Really fascinating work and really interesting system.